Thank you so much for having us. I'm Claudia. I'm a postdoc in Brian's lab, and I was working with this fantastic team of PhD students and undergrads to work on this model. I'll let Talal introduce himself.
Yeah, I'm Talal, PhD student in Brian's lab, third year. I work on protein design in general, a lot of work with inverse folding models, trying to get our generative models to generate proteins that we want to use and practice in the lab. Yeah, and this project was really fun. It started with, a conversation between me and Santi, and then John, an undergrad I was mentoring. Uh, Jumped on, and him and Santi ended up really carrying the computational work of the project. And then we had Claudia Hauyu and Zhao doing amazing work experimentally, and it was just really fun for everyone to come together and work on this, and really excited to share the story with you guys.
I think Brian
Awesome.
described the team as a lean and scrappy team, and I think that describes it perfectly. Um, anyway, so we're going to be talking today about one of the many very exciting antibody design models that came out over the last couple months. Ours is Germinal. I'll start with, the obvious question, why antibodies? Despite, being at the core of innate immunity, playing absolutely vital roles in mammalian immune response, antibodies are both clinically and economically hugely valuable for use as therapeutics in viral neutralization, antibody blockade therapy for many cancers. Antibodies also play a role in signal activation and are particularly potent as carriers for drug delivery and other delivery mechanisms. But if anyone's ever, stepped foot in a lab, they probably will have used an antibody for, research application, for example, ELISAs or IP or in flow cytometry or imaging. And as we kind of have entered into more synthetic biology space, antibodies have become particularly important for uses as biosensors or for synthetic receptors for uses in chimeric antigen receptors in CAR T cells. The kind of traditional and historical model for or approach to generating a new antibody towards your antigen of choice or epitope of choice is traditionally human or animal immunization. So, injecting your mouse or your llama, in the case of nanobodies, with the antigen that you're interested in raising your antibody against, waiting many months, and then, extracting plasma and isolating the sequence of the broad repertoire of antibodies that are raised against that antigen. And you're then kind of stuck in a position where you either have to humanize that antibody that's been generated. You've either raised your antibody against, not your ideal epitope, or potentially against a target conformation of your antigen that was undesired. Particularly in the case of immunizing mice, you're raising your antigen against differentially glycosylated forms of your antigen. But kind of broadly, you don't have much control over the epitope you're targeting. This is a similar story for raising antibodies or single domain antibodies using display systems. The most prominent being phage display, but people will be familiar with yeast display or mammalian display, where again, you're generating a very large randomized library, or it can be targeted library of diverse sequences, and you're basically iteratively panning over in many cases many rounds to identify binders that bind your desired target. But again, you're not really targeting a specific epitope, but more a protein that's able to be displayed on a surface. In this instance, you're biased by how you've designed that library. Different randomization library approaches generate different biases in protein libraries. And again, you're in this position where you may require multiple rounds of affinity maturation once you've found your lead binder.
Yeah, thank you, Claudia, for giving a good background to traditional antibody discovery. So leading up to this work, we noticed a sort of rise in the ability of epitope-specific de novo binder models, specifically ones that are now able to achieve very high success. Um, at the time of when we were working on Germinal, it was sort of RF diffusion, RF antibody, alpha proteome, and Minecraft. But obviously now and in the past month, it's been amazing. You've seen all of these models, Chai 2, Jam 1 and 2, and also Boltzmann, also able to conquer this de novo antibody design task, which is honestly quite amazing. At the start of my PhD, this was a very, very daunting grand challenge. I thought it would take much longer to occur. And now, 2, 3 years in the future, we have these models which can, you know, um, including Germinal and these newer models, design antibodies within sort of a well-played scale. Um, oh, you're changing the slides. Okay. Um, okay. Okay. I'll just simulate real time. So yeah, so most of these models fall under two different frameworks. So we either have hallucination from a structure prediction model, and briefly how this works is you're essentially inverting your structure prediction model. So AlphaFold is a model that takes in sequence and then predicts the structure. So we're actually going to invert this. Um, and update the sequence with respect to AlphaFold's confidence in the structure, and use that gradient to iteratively update our sequence until we find a sequence which AlphaFold is very confident about. And it turns out that if AlphaFold is very confident in the sequence, it's very likely to be stable, express well, and work in the lab. And that generalizes also to binders in terms of binding their target. And then on the other side, we have a structure generation via diffusion. And briefly, this works as iterative denoising in the case of diffusion models from isotropic Gaussian distribution to clean data. A good example of hallucination would be Minecraft in terms of binder design, and for diffusion would be Alpha Proteo, which we suspect to be diffusion, but I guess the model still hasn't been released yet. Oh wait, sorry, go back. Yeah. And as I mentioned at the time of this work, Chai 2, Jam 2, and Bolshan either hadn't come out or were still coming out, and really the only model that had done de novo binder design was RF antibody. However, if you compare it to RF diffusion, Alpha Proteo, and Minecraft, the success rates were still very, very low. Um, and if you go to the next slide, Claudia. Despite having very low success rates, retrospectively, RF antibody found when using AF3 that you could actually, sorry, when using AF3 on the binders generated by RF antibody, they found retrospectively that AF3 confidence can predict binder success rate very well. Um, so looking at the antibody design task, this diffusion model was not necessarily able to achieve high success rates, but we see that with this filter, the success rates can be boosted. So then the question comes to mind, why not directly optimize for this filter? Especially on the heels of Minecraft, which saw very high success rates only by optimizing for um, structural confidence, and of course having many filters on top of that. Yes, Hannes.
So the argument would be a little bit because I want both. I want to have high likelihood under a generative model and to have high IPTM, not just to have high IPTM.
Yes, and that is sort of our motivation for also using IGLM in our approach.
Ah,
Um,
now we have high likelihood under a sequence generative model. Makes sense.
yes, and then yeah, and then so I sort of agree with the whole premise that you don't only want to directly optimize under your structure prediction model. And I also think that's why in Minecraft it was important to do that last sort of design step with soluble MPNN, since the structures and yeah, if Martin wants to chime in, we have the
Yeah,
creator here.
that would be
Um,
amazing, Martin, if you share your views.
Yeah, hey, great work, guys. I'm really glad to hear this. Yeah, the soluble MPNN thing actually that came from another project we were doing previously with Casper Goverda on solubilization of membrane proteins. Like we've done a lot of hallucination designs there, and we've tested a lot in the wet lab. And basically, we've tested a lot of different sequence design things. where we figured out that was actually before MPNN became like very popular, and we met with Eustace early on to train a soluble version because the vanilla one was just redesigning everything back as a membrane protein. But anyways, we found out that basically just the hallucinated sequences most of the time were not either soluble or not even expressible for whatever reason. Because if you look at the structures and sequences, they look pretty normal, but for whatever reason they just didn't. And you really needed this. As a side note, the learned potential pipeline from Posu Huang's lab worked just as well as the MPNN. It was just a lot less computationally efficient.
What is the learned potential pipeline?
It was like a Rosetta-based thing where instead of using the Rosetta function, they had like a trained model to guide the sampling. It was from I forgot the
Anamrata.
name of the first. Yeah, exactly.
Okay, so you sort of have make mutations in an MCMC fashion to optimize this thing?
Yeah, pretty
Okay,
much.
cool. All right, thank you. And Talal, if you
So,
have any
so
comments
yeah,
or
yeah.
continue.
So as Hannes mentioned, and as Martin chimed in, the sort of higher level lesson is that AlphaFold optimization is great, but at the end of the day, you need some bias from some generative model. In the case of inverse folding models, that can be your P of seek give instruct, or with IGLM, as we'll show later, just a general P of seek to actually have these designs actually work out in the lab. Next slide. Um, yeah, perfect. Yeah, and there are some trade-offs between diffusion or generative model and hallucination. So on the left side with hallucination, one of the advantages is that designability is essentially free. And for those that don't know, in the community, when we refer to designability, we generally mean, say you have a diffusion model that outputs a backbone, and you inverse fold this backbone into a sequence and repredict the structure. Is the predicted structure going to recapitulate the initial backbone? Um, or for a sequence, is the sequence designable? Will AlphaFold predict the sequence with high confidence? And since this we're explicitly optimizing for AlphaFold confidence, we sort of get designability for free. In some way, this is sequence structure co-design since we're updating the sequence based off of the predicted structure. And also, your target is flexible. As you do these rounds of AlphaFold optimization, your target actually might move to accommodate the binder you're designing. The main disadvantage is that it's extremely slow. And as we've already mentioned, it can overfit to the objective and get some adversarial generations that might not actually work out in the lab. Um, there's a bit you could ignore sort of that gamma parameter, that's sort of a legacy thing. But yeah, the advantage of the fusion is that it's much more fast. But the disadvantages, as we've seen with our antibody, without any filtering, you will get lower success rates. Next slide. Um, oh, okay. Uh, wait, Claudia, can you go one forward? Okay, here we go. And then additionally, we saw this paper by Del Alamo, um, which used a slew of models to try and recover a binding for trastuzumab. So in this paper, using protein MPL and alone struggled to recover binding given a clinical framework. However, when you actually summed the antibody language model, in this case Abling's logits with the inverse folding model, you're able to get recovered binding. As you can see on that plot on the right, the binders that were in the nanomolar range were all from protein MPN plus Ablang, while using protein MPLN alone resulted in lower binding affinity. So in this case, the antibody language model is acting as sort of a germline or like naturalness bias. And you can also see from these plots here, in terms of just a classification setup using protein MPNN and Ablang was superior to the other models. And next slide, Claudia. So then this begs the question, can we use antibody language models as sort of our antibody-like proxy to guide design? And as Hannes was mentioning earlier, also getting generations that are high likelihood under some generative model and not only biasing towards an AlphaFold multimer hallucination. And one more, Claudia. Yeah, so this is sort of our nice little visualization. So say you have your AlphaFold multimer confidence landscape and you have your IGLM confidence landscape. Sort of The aim of Germinal is sort of to optimize under this joint landscape where we can find designs which are optimal under both models. Yeah, you can go one more slide. So this is sort of the framework of Germinal, where we have our two models, IGLM and AlphaFold Multimer. Um, for the most part, we follow Bindcraft and the whole collab design process with one exception. So instead of going from logits, softmax, and one hot into semi-greedy, for some reason we found in practice that removing the one hot stage helped us get more passing designs. So in terms of stage zero, in terms of where we start, we initialize the framework to some known, very experimentally tested framework with a strong sequence bias. So during design, it is possible to deviate from the framework, but we have a strong bias towards it directly applied to the logits, and we freely design the CDRs. And then the first stage of the design pipeline is sort of the logit representation. So we have a logit input to AlphaFold Multimer. To this day, I still do not understand why logits work with AlphaFold. The model was trained to predict structures given one hot sequences, but somehow you're able to input logits and a structure comes out. Um, and then we slowly anneal to a softmax stage. You can go next, Claudia. Right. So in the logit stage, it's the values are completely unconstrained, but then in the softmax stage, for a given position, all the residue probabilities must sum to one. And this sort of anneals us to a one hot stage. You can go to the next, Claudia, where we have a sort of greedy sort of Monte Carlo search stage where we will sample mutations, evaluate with AlphaFold and IGLM, and then take a mutation if it sort of increases AlphaFold confidence and IGLM likelihood. Um, Right. And we actually see that using IGLM within Germinal steers our generated designs towards natural antibody-like properties. So for this analysis, we use the therapeutic nanobody profiler, which comes from the Dean Lab. Essentially, how this profiler works is they take essentially all the clinical antibodies, and then they sort of look at very key properties, and then are able to come up with thresholds for low, medium, and high risk. So in the three KDE plots on the left, we see the difference between no IGLM and a weighting of one for IGLM. And you can see for surface hydrophobicity, it doesn't make much of a difference, but there is a noticeable distributional shift in the sort of negative charge of the designs as well as the positive charge of the designs. And you don't want these physiochemical properties to be too high. And then on the right, we look at the humanist score. This comes from the OASIS model, which is essentially scanning for nimers in the sequence and computing the similarity to the observed antibody space, which is a large database of antibody sequences. And we see as we increase the weight of IGLM, we get better humanists in our designs.
Can I ask a question in between?
Yes.
What was the IGLM trained for? Trained with? What type of sequences?
IGLM was also trained on OAS.
But
Uh,
what type of sequences are these? Are they mouse? Are they human? Are they humanoid?
Oh, okay. Yeah. Oh, yeah. So it was trained on a range of sequences, and they have like a species label. So you can either condition with human, or mouse, or camel. For nanobodies, because we were using a camelid framework, we conditioned with camel. When we design SEFVs, we condition with human.
Okay.
Will we get into, or like, do you have an overview of all the experimental data that shows us like humanness of your designs, or maybe also
Mhm.
expression ability of your designs with and without this IGLM stuff?
Yeah, unfortunately, experimentally, we did not test IGLM versus no IGLM. We had, sort of a large budget of designs to test, and we wanted to do as many targets as possible. We do have expression data. Unfortunately, it's
Ah,
very hard to get humanness data. I think you'd need to I don't even know what experiment you need to do. You need to put it in a
So that's amazing. What do the expression data look like?
yeah. So I think I have maybe five to
Coming up?
10 more slides, and then Claudia will go
Awesome.
over all the
All right,
experimental
let's wait
data.
for it.
Yeah. Um, okay, yeah, so I think might need to speed up a little bit. So unconstrained hallucination. Essentially at the start of this project when we had no Minecraft and when we had no IGLM and we had no sort of custom losses, we sort of get these sort of designs that really do not look like natural antibodies. Um, this sort of makes sense if you understand what AlphaFold confidence may prefer. AlphaFold really would likes to see secondary structure on secondary structure interfaces, good shape complementarity, and no exposed regions, whereas antibodies will actually bind with their loops. And having loops in an interface will, of course, lower your interface confidence metrics. So, sort of, if you just use hallucination, you sort of get these framework binders. Um, but what is interesting? Okay, I didn't realize Jam had framework binders as well. So, the what is interesting is these framework binders they definitely will bind, but there are reasons we can go into later why this might not actually be sort of the sort of the best binder you want. Uh, um, it if you use sort of a fixed framework and your binder is binding with your framework, it's really hard to argue that the binder will be target specific. Whereas if you use loops with a fixed framework and you're able to design the loops to bind the target, then you're sort of more ensured, at least at a high level, or at least this is our thought going in, that you could get a specific binder. Um, Next slide. In addition to the framework loss, we also use two secondary structural losses. Next slide. But first, going over the paratope loss, which is also sort of the framework loss. You can go to next slide. Sorry, next thing. Yeah. This is just some in silico results showing that when we use no framework loss, we get sort of this sort of uh, a framework binder, as you can see on the bottom, on the right of the KDE plot. And as we turn up the weight, we get more loopy binders. And this also will show itself in other computational metrics which we use as filters.
Talal, have you also
Yes,
just
Warren.
simply tried reducing the definition of the contact loss? Because right now, Colab design just optimizes maximum number of contacts, and you can actually just decrease that.
I think that is something we have tried. I think specifically Santi, so I'm not aware, but we definitely have played with that parameter. But I believe we had to actually explicitly use this paratope loss to not get the framework binders, or we call them sandwich
Yeah,
sandwich
yeah.
binders. Yeah.
Thanks.
Um, in addition to optimizing these sort of framework binders, raw optimization of outfold confidence, you also do not get loop-like CDRs, and you'll get a lot of alpha helices and beta sheets in your CDRs. So we actually borrow the helicity loss that's implemented in Minecraft and apply it to CDR residues only, and we also implement an analogous beta strand loss. And you can see on the right, in practice computationally, using these losses will increase the proportion of loops and decrease the helix and beta sheets. Um, and you can sort of see a visualization here with the binder sort of on the top left having a very large helix proportion, and then on the lower left being more loopy. And we see we're able to modulate for reduction of alpha and beta in the CDRs. So I went over the design stage previously. So we have three stages. So the first stage is our design stage where we co-design with Alfold, Multimer, and IGLM. And then similar to Minecraft, we have a sequence design or inverse folding stage with AB, MPNN. And then our final stage is filtering where we predict with AlphaFold 3 and also calculate Rosetta-based metrics. So this is sort of how I think there's one more, Claudia? No? Step four, yeah. So this is how an average campaign will look like. In this case, PDL1. So you start with your epitope selection, and in this case, there is sort of a natural wild type binder which we actually want to compete away with our design. So we chose that epitope. If not available, this is actually advice from Martin's Boston protein design a talk. We look for hydrophobic patches as epitopes. Additionally, sites with no PTMs. As if there's a PTM, then and it's not being actually modeled with AlphaFold, then that's not actually the hotspot they'll be binding in practice in the lab. And then additionally, we do recommend actually tweaking some of the design parameters, which we go into more info on our GitHub repo. And then after that, after you find some parameters that are producing some success, we do large scale sampling. We're very fortunate to have a lot of, we call it preemptible compute at ARC. This means these are the GPUs that people aren't using, you can sort of use for free. So these are like the background GPUs. So for 3 days on 24 to 36 GPUs, and this is a lot of compute, we understand. This may not be accessible to a lot of labs. We ended up with 20,000 total trajectories. And from that, we got 1,500 completely passing designs. So in practice, you actually do not need to sample that much. We sort of oversampled given this is our first experiment. But essentially, you maybe only need, I would say, maybe 300 fully passing designs that you maybe want to filter down to 50 for testing. So you could probably sample with sort of 1/5 of the GPU hours here. And then the next step is to do sort of your final filtering and ranking. I think, yeah, you can look at the GitHub repo or the paper for all the filters we use. But we ended up filtering down this 1,500 to a final, I think, a final set of 100 for PDL1 for experimental testing. Oh yeah, and these are the filters we use. So there's a combination of structural confidence metrics that come from AlphaFold 3, also IGLM log likelihood, and a bunch of metrics that come from Rosetta.
Okay, thank you so much, Talal. So, everyone loves to see experimental validation. We decided to kind of put Germinal to the test across 4 diverse protein targets. So we started with PDL1, as Talal mentioned, just because it's a clinically relevant receptor. It's also been the target of many previous binder design efforts. And so it was kind of an ideal substrate for benchmarking our model against others. We also opted to design binders against the 2 cytokines, IL-20 and IL-3, which play a role in human immunity. And importantly, at the point of, and I think still today, there were no known de novo binders to either cytokine. So these were kind of a more challenging design task. For BHRF1, this is an example of a non-human antigen. So this is a BCL2-like anti-apoptotic inhibitor from Epstein-Barr virus. And so this was kind of like an example of a viral antigen that we can use as a design substrate. We wanted to be able to test a good handful of designs in a high throughput and very cost efficient way without having to express and purify every single binder and then biophysically characterize them by BLI or SBR. So we opted for this kind of scalable and plate-based split luciferase assay, which I'll describe just very briefly. It basically takes a nanoluciferase, which is split into a peptide component and a large protein component. The large protein component is called large bit, and the peptide is called small bit. Very straightforward. So we transiently express all of our binders fused to the large protein component, large bit. We literally just take supernatant directly from cell media 2 days after expression, and then combine it with either, so just looking at the top pathway now, a version of this high bit peptide, which binds with very, very high affinity to the small bit or the large bit component. And we use this, and upon reconstitution of the large bit protein and the small bit peptide, you reconstitute the nanoluciferase, which then luminesces, and you can use that as a readout for how much binder is present in your solution. So we use this kind of the top component of the path of the pipeline to quantify expression. Simultaneously, we take the same supernatant and we add a version of this peptide, which is engineered to have a very, very low affinity. So while high bit binds with low nanomolar affinity to large bit, small bit binds with like hundreds of micromolar. So you're only going to get reconstitution of small bit and large bit if you have something dragging the two together. Yeah, sorry, question.
No, okay, it just makes a lot of sense. So you have these two things, one thing attached to your target,
Right.
one thing attached to your binder, and they only shine or fluoresce if they are close to each other.
Right, right. And if anyone's worked with luciferases, like that's not, in practice, it's not a perfect kind of on-off switch. You know, you do get some background, but you can account for that with appropriate controls. And so, yeah, so for this bottom binding assay, we simultaneously add binder large bit to on target. So for example, PDL1 fused to small bit, or an off target protein fused to small bit. And so we can use these two luminescence quantifications to measure kind of on target versus off target binding.
Could you also now attach the large bits? So is it a large bit? Whatever is attached to the nanobody. Instead, attach this to the native binder and then see if your binder displaces the native binder.
Yeah, competition. Yeah, we did do some competition assays. We actually didn't include that data in the manuscript, and we don't have it here, but we were using that. Yeah, we were using that approach. You can also do it the other way around, where you can kind of outcompete your you know, for epitope studies, you can kind of make mutations in the epitope and see whether or not you can outcompete binding
Yeah, okay.
of your
I thought
nanobody.
that would make sense if you're actually like if actually the downstream goal was to displace this thing and to
Right.
just directly test displacing the thing.
Yeah, of course, if you have a control that binds the same epitope that you've designed towards, which is actually, we only had that for PDL1 and BHRF. We didn't have that for IL-20 and IL-3. So yeah, so we took you know, luminescence measurements at 2 time points. We'll just call them T1 and T2. But the idea here is that if you have a true binder, you should get an increase in luminescence between your 2 time points. And I'll talk about kind of filtering parameters in a second. But you know, as a whole, we screened like 196 well plate basically, or a bit more for each target. So the important thing about this split luciferase assay is that both high bit signal and PDL and small bit signal scale log linearly with the concentration of your large bit in solution. And this is important because it means we don't have to normalize every single well by the concentration of binder that's in the well. You can kind of post hoc normalize for concentration of binder. So we first performed the expression assay. So this is just adding large bit binder plus high bit peptide alone. And this quantifies expression of your binder in solution. So we see kind of you know, a range of expressions, but we actually experimentally set this threshold of everything in gray was filtered out as a low expressor. And we experimentally set that threshold by whether or not the binder could be expressed in XB293 cells. So the luciferase assay was performed in HEC293s, and if it could translate to XBs, then we set that as like a true expressor. So we filtered out everything that didn't express. We then took those, or had low expression. We then took those well-expressing binders and quantified binding using the small bit assay. And I'll talk about filtering in a second, but we were then able to normalize that binding, raw luciferase signal by expression. And so you kind of have this normalized signal for binding. So we actually empirically determined this filter for the binders based on, as I mentioned before, an increase in on-target luciferase between the 2 time points that we measured versus off-target. So that just means if your luminescence has increased when you add your target bound to small bit, but it doesn't increase when you bind your off-target protein bound to small bit, then we kind of take that to mean it's a true lead. And everything shown in green was then, or everything colored was taken forward experimentally to be quantified by BLI. Everything shown in green was found to be a true hit. So we are kind of picking up these false positives, but that's kind of a better scenario than losing binders due to false negatives. So as I mentioned, we then took all those filtered designs and expressed them all in a higher scale and purified them and then quantified binding by BLI. This is just in the case of PD-L1. We get kind of a range of affinities from micromolar to kind of hundreds of nanomolar. We performed this for all the binders and were able to extract true hits based on kind of a much more stringent threshold for binding by BLI. We then wanted to kind of probe the epitope, so whether or not the designed hotspot is real. So just in the case of this PD-L1 binder F10, we kind of looked at the interface, identified 3 important interface residues in the PD-L1 epitope, mutated them to an alanine. The hope here is that if these residues are important in binding, mutation to alanine should be disruptive or completely ablate binding, which encouragingly we did see to be the case. So for the isoleucine, tyrosine, and glutamate mutations to alanine, we see at least a 10-fold reduction in binding affinity for each of our PD-L1 binders. And in many cases, binding was kind of, literally just a flat line, so below the detection of the threshold of detection for BLI. I want to just, I think I have the curves. I want to just point out that this assay was quite nice because it allowed us to kind of identify, nuances in the paratope residues, so the residues in the binder that contribute to binding. Where for F10, we see a complete obliteration of binding for the isoleucine to alanine mutation because we have this hydrophobic patch on the binder that's kind of driving this interaction. But in the case of E4, so we're looking at these plots on the right, the isoleucine to alanine mutation did not have as much of an impact because we don't have the same patch. Yes, sorry, question.
So I'm not a biologist, so I'm asking you as a biologist, how much would you care about or be interested in now mutations in the binder versus mutations in the target that disrupt binding?
Yeah, this is a good question. I think that we opted for mutations in the target because the designed binder was the thing in question, right? We didn't want to modify anything about the design that we're kind of probing. I think you should, people do paratope mutations as a mode of disrupting binding all the time. So I think this is like a very standard thing that we could have done. But I think as a more kind of direct question of whether or not this is binding our epitope. And I should say, like, these were specific hotspot residues that were used for design. Then yeah, I think like that's why we opted for mutating the epitope rather than the binder.
But it now tells us that the thing you designed, it actually binds like you designed it. Right.
You can't say without structure, but this does indicate that yeah, we're binding the designed hotspot. I will also say, so for PD-L1, as I mentioned, we had a positive control that binds the exact same epitope as the one we designed to, and we saw. Oh wait, I have it in here. For the positive control, we saw the same thing. So you're seeing like at least 100-fold reduction in affinity when you mutate these epitopes for the positive control versus ours, which was encouraging because yeah, there should be an overlap in hotspot. Yeah, so we repeated this for the 3 other targets, and we were able to extract at least one binder with appreciable affinity for each target. With everyone loves to quantify hit rate, so with an overall hit rate of 4 to 22%.
Then
Yeah.
here again, how much do you actually care about the hit rate? Is it more important to you to sort of try to optimize or let's say you have your ranked list, and now you have the top 5 ranking ones, and you could test those, and then maybe your hit rate will be 4 out of 5. But then instead, you now test 5 diverse ones with some additional diversity that then maybe are ranks 1, 5, 10, and so on.
Yeah.
And then you get a lower hit rate, but the highest affinity then actually is higher. So what would you prefer then?
Yeah, I think it completely depends on the application, right? Like if you're just trying to find a binder, you don't care too much about affinity, or you are willing to affinity mature post selection, then just screening your top 5 or whatever is sufficient. But as you say, the top 5 doesn't necessarily mean the highest affinity. And so if you are kind of optimizing for affinity, then it's worth screening. It's worth screening more. But again, like in our hands,
I don't mean screening more. I mean screening the same amount, but screening a
but
more
a higher
diverse
diversity,
set.
sure, higher
Okay,
diversity.
I see. Yeah.
But yeah, I think, and it completely depends on resources available. I think like the plate-based luciferase assay is so easy that if anyone wanted to screen, a whole plate of designs, that's the same amount of effort as screening 5. But if you are expressing each one individually, that's a completely different question.
You say it's so easy. To me, you're still performing half magic.
You can transfer liquid from one thing to another. It's easy enough.
Okay.
So I just wanted to note, I said we did this on off-target versus on-target binding test when we did the binding experiment with the luciferase. The luciferase signal is very, very dependent on target. And so, because it depends on the positioning of the small bit peptide relative to the epitope. And, there are many kind of factors that play into the actual magnitude of that signal. And so you can't just take a raw readout of luminescence to mean whether or, to tell you whether or not it binds preferably to your target versus your off target. And I just wanted to highlight that with, this plot on the left just shows you the darker color is the time point 1, the lighter color is time point 2, and this just tells you like the raw luminescence signal at those 2 time points. And, just by eye, it looks like, okay, well, things are actually binding both on target and off target. So we wanted to, absolutely make sure that's not the case. So we selected, for 3 of the targets, we selected 2 binders, one that had a high on over off target binding ratio and one that had a low on over off target binding ratio. And we quantified binding by BLI. So we purified those proteins, tested binding, and we can see in each case, the binder that had the kind of low on over off target binding ratio was actually found to be a non-binder. And so all this to say is the Lux data alone isn't sufficient for quantifying kind of specificity, and we're currently doing some pretty in-depth polyspecificity assays to actually quantify polyreactivity of our binders. Okay, so we performed how you perform differential analysis on 2 groups within our data, either expressors versus non-expressors or binders versus non-binders, to extract features or metrics of the binders that were found to be statistically have a statistical significant difference between the two groups. So, a higher G means that that value or metric is higher in group 1. So group 1 here being expressors or binders, and a lower G means that that value is lower. And this was just encouraging because kind of, I guess, metrics you'd expect to pull out like a low interface DG or high interface hydrogen bonds were being pulled out from these analyses. We then went on to kind of repeat the design task with SCFEs. So in this case, this is a variable light chain directly fused to a variable heavy chain. I'm sure everyone knows an SCFE, but this is kind of the binding component of an IgG antibody. And so just looking at PDL1 and IL-3, we found it encouraging that our filtering metrics were, yes.
Okay, then let's go do it now. So Chai now, for example, is pretty happy that they can design whole antibodies, so to say, where so you say everyone knows SCFV. I'm not so familiar with the stuff, but now I sort of know that SCFV are sort of these two fused chains that you're showing here that are close together, and then we have this concept of fabs where we then have essentially two more chains or two more domains, like similar to what you have here, and they're not fused together but just bound together. And then you can have full IgG, is that what you call it? Antibodies?
Okay, two things. So so if we take a full IgG, the IgG is composed basically of two heavy chains and two light chains. And the binding component of the IgG is just the light chain fused to like a component of the heavy chain. And as you said, fabs, like the light chain and the heavy chain, they're only covalently bonded by a single disulfide bond. And so to kind of make your binding component, and the thing that drives binding in both in the fab, so the heavy and the light chain, is just kind of the variable regions at the tip. And so half of the fab is kind of constant. You have different constant regions, but it's pretty much conserved across all IgGs. Whereas the thing that actually varies is the variable region. And so the SCFE is literally only the component of the fab that varies. And as you said, you directly fuse those two chains because there's no disulfide bond that pairs them together. That disulfide bond occurs elsewhere in the fab. But I will also say that anyone can correct me if I'm wrong, but I actually believe that Chai did what we did, where you design on the SCFE and then you reformat the SCFE into an IgG for screening. And yeah, in the same case here, we designed as SCFEs, but we actually screened them as fabs because this is kind of a much more clinically, basically converting an SCFE to a fab is shown to not, in many cases, not preserve binding. And so it's kind of the more therapeutically relevant format to be screening in fab format or full-length IgG versus SCFE alone.
But why do I want IgG instead of, why would I put IgG into my body instead of SCFB?
Well, SCFEs, sure, SCFEs are useful for things like bispecific T-cell engagers, so small kind of fused bispecifics. But SCFEs are very, very limited due to their size. So they have a very short half-life in patients. There's like a lot of people, there's a lot of efforts to extend the half-life of SCFEs or SCFE fusions, either fusing it to an Fc domain or like human serum albumin or something to extend the half-life. But they're like passaged through, they're filtered out of the body very quickly, within hours, whereas a full IgG is within days.
Okay, so it's not about being more stable and getting less degraded, the IgG, but it's about being filtered out.
That, and also, you don't have the avidity effect that you get with full-length antibodies, which have 2 binding arms as opposed to just 1. So in many cases, antibodies are favorable because they are able to actually, bridge 2 epitopes or bind.
But that would only be, wouldn't that only be like stronger affinity, so to say, if you had
multiple.
Yeah, if your target has the multiple binding sites in the right distance from each other.
Or like, you're targeting a receptor and that receptor is expressed to a high level on the surface of a cell, right? Then you can like bridge many, and this is kind of how antibody blockade therapy works, which is you just end up bridging or cross-linking receptors at the cell surface.
I see. Thank you so much for.
No, of course, that was a good question. Thank you. So favorable metrics comparable to nanobodies for SCFEs, and we partnered with the fantastic Adaptive to screen these directly in SBR by SBR, and we were excited to see our binders showing low nanomolar affinity. So we're looking at like 30 to 100 nanomolar range across 23 of the 48 designs screened, which was very encouraging. And again, I'll just mention these were screened in Fab format, not in SCFE. So it's good for us to see that we can design these as small SCFEs, but they can translate to therapeutically relevant molecules. So just for sequence novelty, we use MMSeq to align our these are just on the hits. So this is not on all of the. Is that correct, Talal? Just on the hits, not on all passing trajectories.
Yeah, these are on the designs we tested.
Yeah, designs. Okay, designs tested.
But yes, not all passing trajectories.
So quantifying novelty across all proteins in the PDB versus the observable antibody space. And in both the top kind of panels show for VHH for nanobody, and the bottom panels are shown for SCFEs. And we see
Is this for CDRs or for the whole framework?
Just for CDRs. Because we fixed the framework, so that would definitely lead to much higher seq identity. And then for structural novelty, we used fold-seq multimer to identify, for these were just for the hits, so the binders that we quantified by SBR BLI, and we found the protein in the PDB with the highest structural identity, which are just shown in gray as an overlay aligned to the target. And we see in each case, we actually see the closest structurally similar protein, I'd say, is this peptide for BHRF, just this helix. But when you actually kind of zoom into the actual residues, we see very, very little overlap, which was encouraging. So just to summarize, sorry if we've overrun a little bit, but we were able to design both nanobodies and SCFEs with favorable binding kinetics. We've established this kind of pre-screening luciferase method, which we definitely encourage people to kind of adopt if they're looking for a quick and cost-efficient way to screen a panel of binders. We've shown for both nanobodies and SCFEs, sequence and structural novelty, and hopefully our epitope alanine mutagenesis assays hold up in structure, which we're working on currently to confirm epitope specificity of the designs. We're not naive to the limitations of this model. As Talal mentioned, it's pretty computationally expensive, but it's reasonable to generate a handful of designs for screening if the idea is just to generate one or to identify one binder. And yeah, I think like an important thing to note is that the configurations do require some optimization for each target you're working with, whether or not that's because of the the confidence of the target. Like if you have an inherently loopy target, it's going to be much harder to identify highly confident designs of the complex. Also, if you don't have a known epitope or a known biologically relevant epitope, it requires you kind of selecting the appropriate hotspot residues for design. And then, I guess inherently the method is limited by AlphaFold multimer structural modeling power. And then I'll just finish by saying a huge thank you to this incredible and talented team of people at Stanford. And it was a real pleasure to present today and also to work with everyone on this project.
Amazing. Thank you. Okay, then I would just monitor if anyone else has questions before I go with my larger philosophical questions. Yeah, Claudia, do you maybe want to? Okay, Eric Sonson, go ahead and unmute.
Hi, yeah, thanks for, oh geez, there's an echo here. Sorry, okay, can you?
It's problematic.
Sorry about that. Okay. Yeah, I was wondering with respect to your on-target, off-target results. It looked like there was some like covariation in the signal there. And I was wondering, first of all, maybe I just missed the result there, but when you tested the things that did have the high ratio, did you test those to the off-target as well, and they were non-binders to the off-target? And also, like, what do you think is the source of that covariation?
Okay, so the top row in each of these cases is the binder that had the kind of the low or the high, or the low on over off binding ratio. So I guess like everything that's in the top panel by luciferase assay was predicted to be non-specific. And the colored line in each case, and everything on the bottom row was shown to be, or it was indicated by the luciferase assay to be specific. The colored line is binding to on target, and the gray line is binding to off target. And so in each case, kind of the gray line having absolutely no, BLI shift, just which is an indicator of binder binding to target. This was encouraging because it suggests, at equal concentrations, this suggests that you are not getting specific binding to that off-target protein.
I see. So in the bottom row, also you have like these are the ones where the ratio of on-target off-target are low, and in each case, like whether or not the ratio is high or low, you still see like on-target specific binding. Is that what we're looking at?
Yeah, other way around. But so like the bottom row is the high on-target to off-target ratio, which means you're expecting it to bind better to on-target versus off-target, and then in the top row is the other way around. But yeah, we're seeing in each case this suggests that you are getting binding specific to your target and not to your off target.
Okay, thank you.
And sorry, what's on the left of this slide?
The left. So this is just the raw luminescence that you read out when you perform the binding assay, which, wait, let me just go back to. So this bottom panel of the pipeline is what we're quantifying in that instance. So it's binder fused to large bit, binding to target fused to small bit, or off target fused to small bit. And so you would expect to see a high luminescent signal if your binder is binding to your target, because it's kind of pulling the small bit and the large bit together. But if you are not seeing any binding, you'd expect to see a low luminescent signal. And so like what all this is showing is, to how high was that luminescent signal when you mix those two components in solution. And in pink, you have like your on-target binding, and in gray, you have your off-target binding.
And the conclusion, I guess, is you can't really measure specificity with that assay, so that's why you have to do the BLI. but it turns out
Exactly.
that the ones that are on target high actually are specific.
Yeah, like when we first started this assay, we thought it would be fantastic if we could just pull out specificity from this assay alone, but it quickly became evident that, it's too noisy. And as I mentioned before, the luminescence is actually very, very dependent on the target construct and, the location of the small bit relative to the large bit. And, anyway, there's a number of like optimization things in the assay that we can now spend time working on, but we didn't at the point of this. So we just kind of opted for testing on BLI.
Thank you. Cool.
But yeah, this is why like a more in-depth polyreactivity assay is like what you see in CHI and otherwise is needed for actually quantifying the polyreactivity of your binders. And this just basically means quantifying binding to abundant proteins in mammals.
What is the polyreactivity stuff that's in CHI?
So they do an ELISA actually on like double-stranded DNA, single-stranded DNA, like ovalbumin, a BVP, just a nanoparticle basically. And these are just like either things that are known to be sticky or things that are highly abundant in humans. So you're basically like trying to quantify whether or not you're going to get some off-target stickiness in the context of where these antibodies could be used.
So what we did with Adaptive was just, and they still had the material for the other designs that they tested, and then they just tested them against some off-targets. That's HSA, human serum albumin, and that was very cheap. So how does this scale or rank on your scale of meaningfulness?
I think that's very, I mean, it's a very important indicator whether your binders bind HSA or not. We are doing a slightly more in-depth assay, which basically quantifies binding to all solubilized surface proteins that exist in mammalian cells. So it's like beyond just HSA, but it's kind of all exposed proteins. But I think the HSA polyspecificity assay is still very relevant and important that people do.
Okay. But it's certainly are like is it fair to say that you care much less about it than like a large polyreactivity test? Or where does it rank, so to say? Is it much better than just testing an off-target like BHRF1?
Yes, it's better than just quantifying binding to like some other off-target protein.
Random, okay.
But I would not be surprised if you did see if you see like stickiness to BH or like an off-target random protein, you would also see stickiness to HSA. I mean, I think I think it's definitely a better indicator because it's so hugely abundant that if your binder is sticking to that, then it's a very bad sign.
Okay, friends of the sun, do we have any other questions in the chat or raised hands? Otherwise, I would have the quick question of Claudia. Is there anything you want to share about your adventures for designing, let's say, multi-specific binders that can handle multiple mutations?
Talal, jump in if you if you want to. I think it's important to define, from what we've seen, it's important to define your hotspot well. And I think that if you're designing, it's a different challenge to be like forcing your designs to a highly mutable epitope versus like the most conserved region you can find within that protein. I think like I'm sure you're in the same position, but like we're challenging ourselves with the harder task, which is like designing to an epitope that is mutating. And I think in that case, both biological knowledge of evolutionary trajectories is important so that you can define conserved residues, but also defining which mutated residues can still be included in the hotspot. So we think it's important that you are designing your binder to still bind a mutated interface and not just the conserved region of that interface. I don't know, without giving away too much.
Yeah, and I guess the thing I might add is even though, we've been working on this for a little while, there still definitely are some unanswered questions as to how the best way to do this is. If you could think of the problem as, maximizing your likelihood to bind over your set of input structures, you don't want to do this to the extent where you're maximizing the likelihood of your design to bind to any structure, and now we have some non-specific
Yeah.
binders. So it is going to be yeah, we look forward to sort of to testing these and seeing how they work out and seeing if there needs to be sort of maybe some other parameter that forces you to that sort of constrains you from going in that adversarial direction of just making something that binds everything.
Yeah. I just love that we're now thinking about these sorts of questions. So,
Yeah, it's exciting we're in
more,
the application stage
yeah.
of this design problem.
How do you approach the maximizing likelihood of binding against multiple things question? Is it you have your inverse folding model, so to say, and then you can try to have some AND operation between your probability distributions? Like one is conditioned on one target, the other conditioned on another target, and then you can combine them.
Yeah, that's a known way to maximize the likelihood of a sequence over a set of structures is to add the logits, which is like the equivalent to finding the highest joint probability. I think this has been done with like protein MPNN for when they do like tide sampling of like a dimer, for example.
Yeah. Do you think that does the trick already, or it's a big issue that we then are getting more and more toward designing something non-selective?
Um, yeah, I mean it's a good question. I mean it is the most like theoretically like principled way. However, yeah, there may need to be some sort of regularization to stop from pushing towards just a completely non-specific binder. If It would be amazing if we had some oracle of non-specific binding, but I used to do work in antibody property prediction, and that was always the most difficult property to predict was polyspecificity. There isn't that much data for it, and it's just yeah, it's super hard to predict. But if we had maybe even some heuristics, like knowing that having a lot of hydrophobicity in the interface, or so certain amino acid statistics in the interface would lead to polyspecificity. Maybe something like that could help, like a bias away
Yeah.
from that. But yeah, it's
Well,
interesting
okay.
question.
Amazing stuff. Any last words that you want to say before we call it a day? That's a no.
Yeah. For those who are trying to run Germinal, we're very responsive on the GitHub repo. So if you have any questions, we're happy to help out. Um, and yeah, I can even leave my email here if anyone needs to reach
Email
out.
in the chat. Very nice. Thank you.
Yeah.
Okay. Then Claudia, Talal, thank you. And super nice stuff. And thanks for all the biology explainers. Thanks for getting me a bit of a better understanding of some concepts and what to look out for when reading papers like this. And have a beautiful rest of your Monday.
Thank
See
you
you,
so
friends
much
of
for
the
having
sun.
us. Thank you
Thanks
so
for
much.
having us on this.
Pleasure. And great work
Bye.
on both, by the way. Bye.
Thank you.